Antigen define a substance that satisfy two distinct immunological properties: the immunogenicity and antigenicity. It is the ability to induce immune response in the body. B cell plus antigen effector B cell, T cell plus effector T cell. Antigenicity, it is the ability of the antigen to combine specifically with the final product of the O2 response, that is antibody of the T cell. The substances that satisfy the first property, that is immune induced specific immune response, is more appropriately called immunogen rather than using word antigen. All the molecules having immunogenicity property also show antigenicity, but the reversal is not. For example, heptane, which an antigenic but have epitope. Epitope or antigen determinant is the smallest unit of antigenicity. It is defined as the small area present on the antigen comprising of few amino acid or monosaccharide that is capable of sensitizing T and B cell and reacting on the specific site of the T cell receptor and in the specific site of the antibody that react with the corresponding epitope of the antigen is called paratope. Haptens are low molecular weight molecule that lacks immunogenicity, cannot induce immune response but retain antigenicity or immunological reactivity, that is, can bind to the specific antibody or T cell. Haptens can become immunogenic when combined with the larger protein molecules called the carrier. Haptens may be classified as a complex or the simple. Complex hapten and simple hapten. Complex hapten contain two or more epitopes that can react with a specific antibody, and the hapten antibody complex can be visualized by various methods such as precipitation reaction. Simple hapten usually contain only one epitope. Such hapten can bind to the antibody, but the hapten antibody complex cannot be visualized as easily. The precipitation reaction occurs require antigen host relationship. Based on the antigen host relationship, antigen can be grouped into two groups: specific or autoantigen, non-specific. Specific antigen, these belong to the host itself and they are not immunogenic. Host के पास खुद होता है. Host immune system does not react with the known antigen, which is due to the exhibiting a mechanism called the immunological tolerance. However, something the self antigen are biologically altered. Example, as in cancer cell, self or foreign type. These are immunogenic or three type: allotype, isotype, heterotype. Allotype are the species specific tissue of an individual in a species contains species specific antigen. Isoantigens are the type of the antigen that are present only in the subset of blood group antigen. And histocompatibility antigen, the histocompatibility are highly specific. They are unique to every individual. Heteroantigen belong to two different species are called heteroantigen. Example, antigen of the plants or animal. Heterophile antigen is a type of protein that exhibits in unadapted species external. Heterophile antigen are type of the heteroantigen that are present in two different species but they share epitope with each other. Antibody produced against antigen diagnostic application. Heterophile antigen can be used in various serological testing. Antibody against one antigen by employing a different antigen which is heterophile. Physics reaction is done by typhus fever. Antibody against secret antigen detected. Paul Bunyan test is done for the here cold gluten test here antibody factor influencing immunogenicity, size of antigen, chemical nature of antigen, susceptibility of antigen to tissue enzyme, structural complexity for foreignness to the force. Size of the antigen larger, अगर larger the size more the potent. Chemical nature of the proteins are stronger. Susceptibility and degradation of the antigen by the tissue enzyme produces several having more polymer made of the at least two or more amino acid or immunocomplex protein containing 20 amino acid with the level 4 structure foreigners to the genetic factor. Different individuals of a given species show different types of the immune response. Optimal doses of administration, route of antigen, site of injection, repeated dose of antigen, multiple antigen factor therapeutic application. Adjuvant. The term adjuvant refers to any substance that enhances the immunogenicity of an antigen. They are usually added to the vaccine to increase the immunity. Example of alum, mineral complement and the mixture of and incomplete suspension of killed tubercular bacillus in the oil. Lipopolysaccharide Gram-negative bacilli, example LPS of the border telepathy action, excellent adjuvant for diphtheria and toxoid discipline. The reason for using combined immunization for diphtheria, pertussis, and jaundice in the form of D beta. Other bacteria, their Mycobacterium bovis toxoid mechanism adjuvant, delaying the release of antigen by activating phagocytic by activating TL biological classes of antigen depending on the mechanism of the induced antibody formation. Antigen is classified as a T cell dependent, T cell independent, and T cell dependent are T cell dependent. They are processed and presented by the antigen presenting cell to the T cell, which leads to T cell activation. The activated T cell secretes cytokine that in turn stimulates the B cell to produce antibody. T cell independent and independent structurally similar LPS capsule polysaccharide structure complex does those dependent immune where no memory present, no antigen present step in slowly rapidly activate B cell, activate T cell, mature in mature cell antibody response to IgM3 antibody of all classes can be produced. T independent antigen, there are a few antigens such as bacterial capsule, flagella and lipopolysaccharide that do not help the T cell and the antigen presenting cell. They directly bind to the B cell and stimulate B cell polyclonal, leads to the increased secretion of the non-specific antibody. Detailed mechanism. Super antigen is the third variety of biological class of the antigen resident in the unique feature of super, they can activate T cell directly without being processed by the antigen presenting cell. The B region of the T cell receptor appears to be receptor of super antigen. They bind, they directly bridge non-specific between the major and competitor 2 of the antigen, and non-specific activation of the T cell leads to the massive release of cytokines known as cytokine storm, which include inflammatory interleukin 6, gamma, interferon gamma, interleukin 6, and alpha and beta. This in turn can activate beta cell for increased secretion non-specific antibody hypergrowing. Example of super antigen are various products of the microorganism behave as a super antigen, the most important being staphylococcal and streptococcal bacterial super antigen. Staphylococcus toxic shock syndrome, exophilic enterotoxin, streptococcus pyogen, mycoplasma, yersinia, enterocolitis, yersinia, pseudotuberculosis, viral super antigen, Epstein-Barr virus, super cytomegalovirus, rabies virus, HIV, fungal super antigen, varicella furfur. Disease associated with super toxic syndrome, food poisoning, cardiac skin syndrome, rare conditions such as atopic dermatitis, Kwashiorkor syndrome, psoriasis, acute disseminated encephalitis.